Well, hello, cell biologists. This marks a few milestones for us, this video. First, it is a major turning point in the theme of the class. We're going to move away from cellular energy and function and focus more on cellular structure for the next 2 weeks. Two, this is the first of our last two videos of the first half of the course, the first unit. So by the end of this week, the end of the next video you'll watch, you'll have all the information you need for your e-report and your mastery check. And third, this is the first of the two videos before spring break and mastery check week. So we're really approaching the halfway point of the course now, which is exciting. And I guess the last little perk is that chapter 11 just happens to be a short chapter in the textbook, which means this will be a short pair of videos. I think this video and the next one will both be much shorter in length than what we're used to. So with that, let's dive in. We're talking about roughly the first half of chapter 11, pages 381 to 391, so only 10 pages from the textbook covered here. And what we'll be discussing is the lipid bilayer itself structurally, and what we mean when we say that the lipid bilayer is an asymmetrical two-dimensional fluid. Talk about some of the different factors that impact membrane fluidity, both in prokaryotic cells and eukaryotic cells. We'll talk about where new membrane components are made, which is the endoplasmic reticulum. And we'll end by talking again about the asymmetry, but in this time, how that contributes to the cell having the overall spherical structure that it has. So each and every cell on this planet, on planet Earth, has a plasma membrane. That's one of the defining characteristics of life on Earth. And this plasma membrane is a very fragile, fatty film, so thin that it can't even be seen on a light microscope. We need an electron microscope to see the structure of the membrane. Has about the consistency of olive oil usually, and so that's what's covering our cells. A cell's membrane separates that cell from the outside world, so we've got the inside of the cell, all of the organelles if it's eukaryotic, and all the molecules, the proteins, the nucleic acids, etc. And then we have the external environment, the extracellular environment, which is markedly different. Of course, if we do have organelles, we have more membranes in the cell that are compartmentalizing those organelles, and the interior of the organelles are even different than the interior of the cell. But it's this demarcation, this boundary, this defining of in here being separate from out there that makes the plasma membrane so important. So these membranes protect the ordered non-entropic biochemistry of life from the constant disorder and high entropy chaos of the non-living universe that lies outside of the cell. So suffice it to say, without membranes, life wouldn't be possible. We need this boundary where entropy may not enter so that we can have this ordered process, this ordered set of processes that we call life. But membranes do far more than just demarking where entropy needs to leave its shoes at the door. Membranes are the gatekeepers of the cell. Membranes, because of their hydrophobicity and their structural components that we'll talk about in just a second, really only allow certain molecules to enter or leave the cell. Typically, what's entering the cell is nutrient molecules, monomers, and other things that the cell needs to be aware of its environment. And typically, what's leaving the cell is waste for a single-celled organism, or proteins and other macromolecules that are needed for the overall health of the larger multicellular organism. So this means that the cell is simultaneously a barrier and a portal. And I love this analogy, right? This is what a membrane is. You're looking at it. It's a barrier, like a wall, but a barrier with portals so that there can be entry and exit. It's the phospholipids of the membrane that create the wall, the barrier of the cell, and it's typically membrane proteins that make up the portals. These are channels and pumps, things we'll talk about next week. Next week. Yeah, next week. So in addition to there being pumps and channels, portals that things can get in and out of the cell, cells also have what you can think of as ears or antennae, receptors, membrane proteins that bind to ligands that were secreted by other cells and give this cell that we're considering information about its environment. When that ligand binds to a receptor protein on the outside of the cell, that message, that information is transmitted into the cell and allows the cell to respond. So the three main characteristics of what cell membrane proteins allow is import and export. That's the idea of pumps and channels, portals through the membrane. The receptors that I've talked about just a moment ago, the idea of receiving information from the outside world and then indirectly transmitting that information inside the cell. Those are amazing processes that we'll talk about deep in Unit 2. And then there's also proteins in the cell membrane that allow for the cell to be modal, if the cell happens to be modal. Allows it to change its shape, deform its shape, and move as a result of that shape change. So cell membranes are also mechanically impressive. They can change their shape, as we just said, when they're moving. Cell membranes can be stretched, they can be flexed, they can grow, they can shrink, they can deform without tearing, and most impressively, they are self-healing. If you were to punch a hole into the center of a cell membrane, within seconds, that hole would spontaneously close and seal up again. All of this because of the simple chemistry of the cell membrane. So really quite impressive what the cell membrane can do. Cell membranes also confer function because of the proteins that cell membranes have embedded in them, and in some way the fats that are also in those membranes. Membranes become distinct and unique from each other, and these distinctions allow these cell membranes to do specific jobs. We've already talked about one cell membrane that has a very distinct set of proteins that allows it to do a very distinct job, and that's the inner mitochondrial membrane. The intermitochondrial membrane can make ATP by the buckets full. We just talked about that. And the only reason that happens at the intermitochondrial membrane is because the proteins of the ETC lie in that membrane and pump proteins out. And then ATP synthase with its proton channel is embedded in that same membrane and allows those protons back in for loose, tight, open. So the membranes give the organelles, give the cell, give membrane domains their function as a direct result of the lipids that they contain, and much more so, the membrane proteins that we find in those membranes. So it's for this reason that the different organelles in the cell have different distinct and unique membranes, because each organelle is dedicated to a different purpose or job, and to do that purpose and job, its membrane needs to be well suited for that purpose, and so the membranes differ. So these membranes are very, very common in a lot of ways. They share an overall basic structure and function, but in the details, in the protein specifically, they differ, giving each organelle's membrane its own distinct identity and its own distinct purpose. So all membranes are very similar at their most fundamental level, but they're different in the details. We'll talk about the differences in details in the context of all the cellular compartments and cellular processes that we talk about in Unit 2. What we're really focused on in this week are the similarities. How are these membranes built in general? And then we'll leave the details for later weeks. So that's what we're talking about today, the remarkable world of membranes, but superficially, generally, properties that are characteristic of all cellular membranes, regardless of where they might be found. So first and most importantly, membrane lipids form bilayers when they're placed in an aqueous hydrophilic environment. The cell membrane is a bilayer. It's made up of two opposed layers of phospholipids. I'm sure you've seen diagrams like this before in plenty of other cases. What we see in red are the phospholipids, which we've already talked about in our macromolecule lecture in this class, but we'll talk about them again in just a moment. So the hydrophilic heads are the circles and the hydrophobic tails are the little lines here. And then in green, we have membrane proteins that sometimes span through the entire membrane. What you see on the left is an electron micrograph. This is an electron microscope photograph of a part of a cell. And this very thin arc here is the cell membrane. You can see how thin it is even in an EM photo. And you can tell even here that it's a bilayer. You can see a very solid bottom line, a very solid top line, and some lighter space in between where the hydrophobic tails are. So this lipid bilayer is what makes the barrier between the outside entropic world and the inside, more ordered world of the cell. And it's this lipid bilayer, this hydrophobic bilayer, that blocks the diffusion of most hydrophilic molecules. If you're water-loving, which is just another way of saying if you have charge, full charge or partial charge, if you're hydrophilic, you're going to have a very hard time diffusing across the membrane. You're going to have to go through a portal. You're going to have to go through a door. You're going to have to go through a protein. Contributing to this is, remember, we're all bags of water. Life evolved in the oceans way, way back when. All of the biochemistry of life optimized for an aqueous environment. When life ultimately went terrestrial and multicellular, the only thing it could do to continue to leverage all of that evolution was to take the ocean with it. So we're about 70% more or less water in our entire weight, like we're 70% of our mass is water. And the salinity of that water is roughly similar to seawater because that's where life first evolved. So we're mostly water, which means these hydrophilic components are dissolved in that water, and these hydrophobic membrane components have to somehow deal with that water. So we, as organisms, are a water-based ecosystem, and this actually gives lipid bilayers some very interesting characteristics and allows them to assemble into the structures that they assemble into and do the jobs that they do with no additional energy needed. We just let the universe's underlying principles and laws guide these phospholipids to do what they end up doing. Lipids, of course, as a class of molecules, has the one defining feature of being water insoluble. That's what links all lipids together. However, the lipids that make up biological membranes are amphipathic. Again, we talked about this earlier on in the course, but I'll summarize it here once again. The membrane lipids, more specifically the phospholipids, contain at least one hydrophobic tail, almost always two, of hydrocarbons and a hydrophilic charged head group that contains those phosphates we've talked about before. And as a reminder, when we have hydrophilic things with charge, water can interact with them quite favorably. Here we see water molecules, pink is oxygen and blue is hydrogen. Water is making hydrogen bonds with other water, as it likes to do. And as long as there's some charge in the molecule, like we have a partially negative oxygen here, water can interact with it. We see the partially positive hydrogens interacting with that oxygen. So this molecule can be dissolved because it has some degree of polarity. It's got partial charges. For something that's completely hydrophobic, well, water can't interact with that. If you don't have charge, you're not speaking water's language, and water can't interact with you. Water can only interact with molecules that have charge of some kind, full or partial charge. So this is a hydrophobic molecule. Water doesn't interact with it, so all water can do is interact with itself around that hydrophobic molecule. And look at the structure here. Look at the order here. This is much less entropic than what we see here, with this kind of chaotic mess of water interacting with the dissolved solute. So this lowers entropy, that makes the universe upset, and so the universe, one of the fundamental outcomes of the universe seeking entropy is to sequester hydrophobic things together and have as little water interacting with those hydrophobic things as possible. That's why water and oil will separate in a salad dressing, for example. So back to our phospholipids of the membrane. These phospholipids have a hydrophilic head group, which water loves and can interact very favorably, and have a hydrophobic tail, or again, most often two tails, which water is actively shunning in its drive to increase entropy, a universal drive that requires no energy, simply the universe seeking to maximize entropy. And again, this is the definition of a molecule being amphipathic, that it has a completely hydrophilic side and a completely hydrophobic side. And it's this single property of phospholipids that allow membranes to spontaneously form and self-seal, all without any additional energy needed. So let's imagine it. We take a few of these phospholipids, these amphipathic molecules, and we drop them in water. Let's do the thought experiment. What do we think is going to happen? Well, the hydrophobic tails are going to be repelled by water, shunned by water, and water is going to try to sequester them together so that it can maximize entropy. So these hydrophobic tails are going to be huddled together away from water. Again, the same entropic forces that drive oil and water separating in a salad dressing are going to push these, in this case, yellow hydrophobic tails to the center. What will very easily and readily emerge on the exterior of this structure are the hydrophilic head groups that have no problem interacting with water. There's no decrease in entropy. Water can interact with and do-si-do and dance with these hydrophilic head groups, and so they interact with water with no problem. They face out towards this aqueous, watery environment, and entropy is optimized. The structure we just created, basically a solid sphere, a solid ball of lipid, where hydrophilic head groups are on the exterior of that ball and hydrophobic tails are on the inside, is referred to as a micelle. And I want to point out here that I am limited by looking at a two-dimensional screen, and so are you. What we're really seeing here is a cross-section of the micelle. The micelle would be a sphere that is covered with a surface of hydrophilic head groups. To see what's going on here, it's almost like we cut that sphere in half so we can look at the inside. But in a typical micelle, because it's round, the center of the micelle would be filled with the hydrophobic tails, and they would not be exposed to the outside environment at all. Most membrane components, not just phospholipids, but also sterols like cholesterol, which we'll talk about at the end of this video, glycolipids, these are lipids that have sugars attached to them, even membrane proteins. They all, to exist in the membrane, have to be amphipathic. They have to have hydrophilic components and hydrophobic components, and they behave very much in the way of the phospholipids we just discussed. But membrane lipids, like phospholipids and glycolipids, have two hydrophobic tails. And what I've kind of been alluding to here is that the examples I've been giving were for one hydrophobic tail. So if you have two hydrophobic tails, especially if one of those tails is kinked and not straight and linear, that rapidly gets to be too much hydrophobicity to fit in the center of a micelle. So micelles only work with short hydrophobic tails, saturated hydrophobic tails, and or single hydrophobic tails. Once you've got long hydrophobic tails, you've got two of them, or you've got a kink in them. A kink, as a reminder from our early lecture, due to the unsaturated double bond that's somewhere in that hydrocarbon chain. Once you've got these properties, you're no longer going to fit in the center of a micelle. And so micelles can no longer form. Excuse me. Yeah, it's great that we have some unsaturated fatty acids in there. That's going to make us more heart healthy. That's going to make the membrane more fluid. That's going to kind of block this hardening of the cell membrane, but it means that micelles aren't possible. So when you do have these properties, two hydrophobic tails, one has a kink, they're long, they won't fit in the center of a micelle, what can these membrane lipids do in an aqueous environment to still maximize entropy? Well, they create a bilayer. The hydrophobic tails still pack together away from water, but now they do it tail to tail. And the hydrophobic, that should say the hydrophilic, now the hydrophilic heads face the watery environment, both inside and outside, because we've made a hollow ball. We've made a hollow bilayer. We've made a hollow sphere where water is inside and outside. What we've made is a liposome. So if we look at it here, now we've got head groups that have two hydrophobic tails coming off of them. Those two tails are long and presumably one is kinked. These will not spontaneously form a micelle because these tails can't fit in the center of a micelle. So when you drop a bunch of these into water, they spontaneously, without any additional energy needed, form the structure that you're looking at here, this hollow liposome. We have an outer surface of hydrophilic, again, this is a typo, sorry about that. You have an outer surface of hydrophilic head groups that's interacting with the water outside favorably. You have a hydrophilic surface of head groups inside the liposome that's facing the aqueous watery environment of the contents of the liposome. And then on both sides and in between those two hydrophilic head groups, you've got the packing and stacking of all the hydrophobic tails. And again, we're looking at this in cross-section. So if the entire sphere of the liposome was here for us to look at, we could easily appreciate that there are no hydrophobic tails exposed to the watery environment at all. Now, take that liposome and add some stuff to it, some organelles, some DNA, some proteins, pack this hollow interior with the ingredients of life, and you don't have a liposome anymore. You've got yourself a cell. So the phospholipid bilayer of the cell has this structure. It's structured like a liposome. A liposome is a hollow sphere of hydrophobic fatty acids along with hydrophilic head groups, and a cell is a not hollow sphere, a sphere that's filled with stuff, but with the same membrane structure. So this again is the way that we're typically showing this in textbooks and in classes. You've got your dark red hydrophilic head groups interacting with water outside and inside. You've got your light red or pink hydrophobic tails packed into the center. It's two tails per head group, and some of those tails are kinked. That's why they can't form a micelle. They have to form this liposome. And so the inside of the liposome contains water as well. No biggie, because the hydrophilic head groups are facing that water as well. This is what a lipid bilayer actually looks like. You can see it's much more chaotic. Once again, in dark red are the hydrophilic head groups, and now in orange are the hydrophobic tails. But we see that it's fluid, it's dynamic, and water is kept outside and inside. And we have this boundary between outside and inside. The watery environment inside the cell is kept separate from the watery environment outside the cell. It's the same general property that also drives membrane self-sealing. So let's imagine that we've made a sheet of this bilayer, not a sphere, but a sheet of this bilayer. Well, since it's a planar sheet, all where we see this white strip, this is all hydrophobic tails here, exposed to water now along the edge. Well, the water around this edge is going to be very ordered as that water tries to contend with hydrophobicity, and that's going to decrease entropy. So what spontaneously happens, simply because the universe is always striving to increase disorder, what spontaneously happens is that this planar sheet of bilayer begins to fold up into itself and will spontaneously form a sphere, because it's only this spherical structure that's going to have no exposed edge of hydrophobic tails. Only in a sphere like a liposome are you going to have all of the hydrophobic tails sequestered away from water. So that's why cells assume this spherical shape, simply so that there are no free edges where hydrophobic tails are exposed to the water. In water, the lipid bilayers must be free of these edges so that entropy can be maximized. The chemistry of lipids and the universe's drive for chaos demands it. So lipid bilayers spontaneously roll into balls to conserve free energy, to maximize entropy. And again, this is why membranes are self-sealing. Let's imagine that we had a little microscopic hole punch, and we punch a hole in this membrane right here, right? And we pop that hole out. We got a little circle of membrane. We throw that away. Well, all around the edge of the hole we just created are exposed hydrophobic tails. And the water around the hydrophobic tails of that hole are going to be ordered. Universe isn't happy. So to increase entropy and to have the maximum amount of entropy possible, the remaining phospholipids, the remaining membrane lipids, are going to be driven closer and closer to each other to ultimately completely close that hole, so that again we have this infinite layer, if you want to think of it, an infinite layer of hydrophilic head groups, because it's never ending, and no exposed edge of hydrophobic tails that water has to contend with. These processes are not energy costing. They don't need to be driven by the cell. They happen completely spontaneously because of the universe's tendency towards chaos. Okay, so let's talk about the membrane being a two-dimensional fluid. What does that mean? Well, membrane lipids contain hydrophobic regions. We've just said that. And these hydrophobic regions can never exist happily and in a fully entropic state in an aqueous environment. At all times, the universe is going to try to cluster and huddle those hydrophobic bits so that none of them, ideally, are exposed to water. Again, it's this idea that the enemy of my enemy is my friend. It's not so much that the hydrophobic tails are sticking to each other. That'll only happen if they get to that magic van der Waals distance. Instead, it's that these tails both are being pushed on and repelled by water, and so they're being huddled together by this combined exclusion by the water in the environment. But it's this reason that keeps membrane components from ever spontaneously leaving the membrane. You can think about it. Imagine we have a membrane. We've got a nice sphere of a liposome. I'm going to go back so we have a visual. And let's imagine that one of these phospholipids tried to emerge from the membranes. It tried to pop out. Well, as soon as it pops out, some of the hydrophobic tails is going to be exposed to the water that's out here. As soon as this membrane component tries to pop out, now some of the hydrophobic tail is going to be exposed to the water. That's going to decrease entropy. It's going to piss off the universe. And with no energy required, that membrane lipid is going to be forced right back into the membrane so that water can't see it. So things can't pop out of the membrane on their own. There's at no time will something ever spontaneously leave the plane of the lipid bilayer. They're quite literally stuck in there by this constant exclusion of water. Now, can something be forced out of the membrane? Sure, of course, but it requires energy to do so. Right now, we need to put energy in to do the work to force this out of the membrane. Left to its own devices, things are going to stay in the membrane and not leave because no energy is being put into that system. Now, all of that said, there's no force that's stopping membrane lipids and membrane proteins from floating laterally in a membrane. Now, they can't pop up to leave the membrane, or these phospholipids can't pop down to leave the membrane, but there's nothing stopping these phospholipids from floating within their plane, very much like icebergs float in the sea. What do we mean by this is floating forwards, backwards, left or right. If this membrane lipid right here were to float forward, no, its phospholipid tails are, I'm sorry, its hydrophobic tails are not going to be exposed to water. If it floats back, same. If it floats to the right or the left, no exposure of the hydrophobic tails. So that kind of lateral diffusion is allowed, and it happens all the time in membranes. It's the norm for membranes. What we don't get are components moving up or down out of the plane of the membrane. And this is why we say that the membrane is a two-dimensional fluid. It's constantly in motion, but only in the two dimensions of front, back, left, right. There's never any third dimension movement where these molecules float up or down in the membrane. Because as soon as something floats up or down, we're either going to have hydrophobic tails exposed to water floating up, or the charged hydrophilic head group exposed to other hydrophobic tails if we try to float down. And both of those are going to instantly be reversed without any energy needed because of the entropy that it creates, the friction that it creates. The thermodynamic friction, not real friction. Another thing that membrane components can't easily do is transversely diffuse. They don't easily flip upside down. Think of this as an iceberg tumbling upside down in the ocean. Any individual membrane lipid or membrane protein typically flips upside down in this way roughly once a month. And if you know anything from biology at this point, you know that that's a very, very slow process. That's a long time for anything to happen in the cell. So this is what we mean by transverse diffusion, by flipping. So here's our phospholipid of interest. It can float right, no problem. Lateral diffusion. It can float left, no problem. It can float towards us or away from us. That's not going to be a problem as well. But if it tries to move up out of the plane of the membrane, these hydrophobic tails are going to be exposed to water. That's going to drive it right back down. And while it can flip upside down and diffuse transversely, it's not going to do that often at all. It's going to only do that, again, about once a month. And this is transverse diffusion, as opposed to the lateral diffusion that we've already discussed. Transverse diffusion screws up a lot of things. We're going to see in just a second, we actually need asymmetry in a cell's membranes. And if things are allowed to transversely diffuse and not be corrected, then we'll lose that asymmetry. Ultimately, the two layers of the lipid bilayer will homogenize and they will no longer be asymmetrical. Might take a lot of time for that to happen, but if this transverse diffusion is not corrected, it will eventually happen. So even though it's rare, we have to fix it. And because it's so rare, we can't count on it flipping back into the right position. If it takes 3 months for this phospholipid to get here, it's going to take another 3 months for it to maybe flip back to its its correct face. So again, this has to be fixed. It has to be corrected. And it is corrected by enzymes that are cutely named flipases. So flipases fix transverse diffusion. Flipases identify a membrane component, a phospholipid, that's on the wrong face of the membrane and actively moves and flips, forces the transverse diffusion of that lipid back into the proper state, into the proper face. Now, all of these discussions we're having on diffusion of membranes, I'd like you to keep those separate in your mind from the idea of membrane flexibility. Membranes can bend, take on different shapes, stretch, and deform with no problem at all. What they can't do is transversely diffuse, right? So flexibility is totally allowed. Lateral diffusion is largely totally allowed. We'll see some rare examples where it's intentionally blocked, but without some active force to block lateral diffusion for a specific purpose, membrane components are going to laterally diffuse without any problem at all. The one thing that can't happen is this idea of transverse diffusion, the flipping of membrane components from one lipid bilayer to the other, from one face of the membrane to the other. Okay, so now let's talk about the membrane's fluidity. How dense or loose is it? Is it more like olive oil, or is it more like butter or lard? When we say fluidity of a membrane, we really just mean its density or its looseness. The more fluid a membrane is, the less dense it is, and the easier it is for lateral diffusion to occur. The less fluid a membrane is, the denser it is, and the harder lateral diffusion becomes. Again, floating around in olive oil versus floating around in butter. Depending on the particular cell and the environment that it's existing in, different fluidities are ideal. Some cells require a denser membrane, and other cells are optimized to have a more fluid membrane. And also, single cells, usually prokaryotes, can often regulate their membrane fluidity. If it's in an environment that requires more protection, it can intentionally make its membrane more dense. And if it's in an environment where it wants to maybe absorb nutrients more readily, it can make its membrane more fluid. One contributing factor for membrane fluidity is, of course, absolute density, how closely membrane lipids can pack together in a single space. And we've already learned something that pertains very directly to that. Looking at this diagram, we can already tell that this is a more fluid membrane and this is a less fluid membrane. This is less dense and this is more dense. These phospholipids are taking up more room, making them less dense. These phospholipids are stacking and packing much closer together, making them more dense. But why? What is it about these phospholipids that's making them take up this room and making them less dense? Well, it's that they have a kink in at least one of their fatty acid tails. So both hydrocarbon tail length and hydrocarbon saturation contribute to membrane fluidity. And by saturation, of course, we mean the number of double bonds, the number of kinks. If you're a saturated fatty acid, you have no double bonds, you have no kinks. And if you're an unsaturated fatty acid, then you have at least one double bond and you have a kink. So these kinked fatty acids, they're going to make less fluid membranes. Another factor that we haven't discussed yet in any detail is that longer fatty acid tails contribute to dense packing. So if you've got 20 carbons or more, you're going to make a denser membrane. And if you have less than 20 carbons, the membrane is going to be a little bit more fluid. And then again, the presence or absence of saturation of these double bonds is going to contribute as well. So what are your densest, least fluid membranes? They are your completely saturated, long 20+ carbon fatty acid tails on your phospholipids. What's your loosest, least dense membrane? Short fatty acid tails under 20 carbons that have at least one double bond in them to make those kinks. And then anything in between, right? If you're long and you have a kink, or if you're short and you're saturated, you're going to have an intermediate density between the most dense and the least dense, the most fluid and the least fluid. And it's important to point out, too, that when you are this closely packed and this dense, you often are at the magic van der Waals distance, and that contributes even more to the density and the lack of looseness of these membranes, because these hydrophobic tails are close enough that they're inducing dipoles in one another, and that stronger attractive force is making the membrane even less dense, even less fluid. So everything we just described pertains directly to prokaryotic cells. Bacteria regulate the fluidity of their membrane in this way all the time. Bacteria have enzymes where they can lengthen or shorten their fatty acid tails and introduce or take away double bonds and kinks. So bacteria are tailoring the density of their membranes to what they need in any given environment based on changing and modifying the length of fatty acids and the presence or absence of double bonds. We don't do that. We can't easily manipulate our hydrocarbon tail length or our level of saturation. And so we need to regulate our cells' membrane fluidity in a completely different way. And we do it through the use of cholesterol. So this is what cholesterol is for us. It is a membrane denser. It is the molecule we use to make our membranes more dense. So this is what cholesterol looks like in the context of a membrane. It has its classic sterol 4-ring structure, which is very hydrophobic. It's got a long hydrocarbon tail, which is also hydrophobic. And there's a little tiny polar head group here, but it is polar. It contains partial charge. So technically speaking, sterol is amphipathic because it has a polar head group. And for a healthy, typical human cell, about 20% of that human cell is made up of cholesterol. So this idea that cholesterol is this horrible molecule that we need to avoid at all costs, nothing could be further from the truth. We require cholesterol to help us regulate our membrane fluidity. If we have cells that require a denser membrane, well, then we'll see that the cholesterol composition is significantly higher than 20%. And for cells that have more fluid membranes, then we'll have much less than 20% cholesterol. So the more cholesterol in a membrane, the denser that membrane is going to be, and the less fluid it'll be. When we, as eukaryotic cells, as animals, have a typical phospholipid, so we've got a nice hydrophilic head, we've got a saturated fatty acid tail and an unsaturated fatty acid tail. And so we've got that kink there. We've got something that's going to make this membrane less fluid. Well, we can't regulate the level of saturation. So if we need this membrane to be more dense, then what we do intentionally, what our cells do, is insert cholesterol in the gaps between the phospholipids, the gaps that are induced by the presence of those kinks, by the presence of those double bonds. Now, if we look, well, we can anchor the cholesterol properly because the little tiny polar head group is going to interact favorably with the charged head groups of the phospholipids. And then the structure of cholesterol with that four-ring sterol structure and that long hydrocarbon tail is going to slip perfectly in the empty space between these two phospholipids. And not surprisingly, perhaps, the distance between cholesterol and these fatty acid tails is going to be a van der Waals distance. So we're going to make van der Waals interactions here between these two and van der Waals interactions here between these two. And so essentially cholesterol acts as a bridge or a glue, gluing adjacent phospholipids together through the adhesive force or the attractive force of van der Waals interactions. And that gain of attraction and filling in of the empty gaps results in a stiffer, more dense, less fluid membrane. So here again is a accurate representation of a dense animal membrane. Again, just to orient you, everything in dark red are hydrophilic head groups of the phospholipids. Here again is orange, is the hydrophobic tails, just like the image that we saw a few slides ago. And everything in green is cholesterol. Look at all the gaps cholesterol is filling. So cholesterol takes up the space between these unstructured, kinked, room-taking hydrophobic tails. And in doing so, cholesterol adds structural stability to the membrane and allows van der Waals interactions to occur. It bridges the distance between adjacent hydrophobic tails, allowing van der Waals interactions to occur on either side. And all of these factors combined make for a much denser, less fluid membrane. Less fluid membranes have a harder time facilitating cell division. So this is one way that cells become quiescent. It leaves cells less motile. Sometimes we want to regulate the motility of a cell. And so we can regulate that motility by adding cholesterol to the membrane. And yeah, I don't want to make it sound like cholesterol is all good. Certainly too much cholesterol is bad. Well, where is that too much cholesterol coming from? Our diet. Where's the first place that consumed cholesterol ends up after it's eaten? In the bloodstream. What cells does the bloodstream come in contact with? The cells that line the arteries and veins and capillaries. And so the cholesterol, the abundance of cholesterol in an unhealthy diet slowly leaches into the cell membranes of those arterial walls. And when we say that arteries harden, we mean it. Because there's too much cholesterol in the cells of the walls of the artery, those cells harden. They become more dense. And what do arterial cells need to do? They need to stretch and contract. They need to help pump the blood. So now you've got a vessel that should be flexible due to a looseness of its membrane, no longer flexible because of the presence of cholesterol. So we've gone from an elastic hose to a PVC or metal pipe. And then unfortunately, at some point, there might be too much blood pressure in that hardened artery. And rather than flex to accommodate that blood flow, the artery ruptures. And you've got either a heart attack, you've got a stroke, you've got some other cardiovascular tragedy. So too much cholesterol, not good, not good, because it does exactly what cholesterol is supposed to do. It stiffens membranes, it adds density, and it makes membranes less fluid. Lots of times that's exactly what we want, but too much of anything is going to cause trouble because you'll begin to make the membranes of cells denser than they should be and need to be to do their particular job. So I hope I've made it somewhat clear that membrane fluidity is critical for all cells. Cells go to an enormous amount of trouble and effort to make their membrane fluidity ideal for whatever that cell needs to do, whether that's the insertion and removal of cholesterol or the regulating of hydrophobic tail length and saturation for prokaryotic cells. The more dense a membrane is, the less lateral diffusion will be possible. We're going to talk in later lectures about membrane domains and creating membrane islands, membrane protein islands. One way we can do that is by controlling the density of the membrane. Membrane fluidity is important for cell signaling. We'll see how and why that is in a couple weeks when we talk about cell signaling. And it allows proteins to get from where they are to where they're going. You need a fluid membrane for a protein to laterally diffuse to its ultimate destination. And if a protein does need to migrate through the membrane laterally, and the membrane is very dense and stiff from too much cholesterol or too much saturation, then we're going to see that that protein is impeded from getting to where it needs to be, and the cell will be harmed as a result. And membranes, of course, are playing a role in cytokinesis, mitosis, cellular division, and we need a fluid membrane for cytokinesis to occur. When that actin belt is tightened around the circumference of a cell to pinch one cell into two, the more fluid that membrane is, the easier it will be for cytokinesis to occur. So lacking membrane fluidity would harm cell health in a lot of different ways, which again is why cells work so hard to keep that fluidity exactly where it should be. So cholesterol is an important molecule, but again, too much of it, not good. So where do phospholipids come from? Where are membranes made? And surprisingly, they're made in the endoplasmic reticulum, mostly in the smooth ER, but also in the rough ER as well. In eukaryotes, such as us, new membrane phospholipids are first made by enzymes in the ER. And fatty acid chains are the substrate, glycerol is the substrate, those phosphate groups, those alcohol groups are all substrate. So literally, we assemble phospholipids from their basic monomer parts right there in the endoplasmic reticulum, in the ER. And then these newly minted, newly born phospholipids are inserted into the outer face, the face of the ER that faces the cytosol, and shown here. So this is just a little bit of the ER membrane. This is the face of the membrane that's facing inside the ER. This is the face of the membrane that's facing outside the ER or the cytoplasm. And these newly made phospholipids are inserted into the outer face. And when you add these phospholipids to the outer face only, exactly what you see in this diagram happens. The shape of the ER membrane begins to deform. It begins to bend because the outer leaf is bigger than the inner leaf. And that's not what the ER membrane should do. The ER membrane should remain relatively straight. So to avoid this, we have a protein that's essentially the opposite of flipase. Flipase moves membrane components, moves phospholipids back to their original face. Scramblase intentionally scrambles the phospholipids of the membrane, goes for uniformity and heterogeneity. So scramblase intentionally forces transverse diffusion to get some of those newly inserted phospholipids onto the other side of the membrane, the other leaf of the membrane, making the number of phospholipids on both leaves roughly equal, and then no bend is induced. We have a planar, more straight membrane, which is exactly what the ER should have. So scramblase distributes these new phospholipids evenly on both faces of the membrane, working around that distortion. Once those phospholipids are in the ER membrane, they can then get to other organelles or even to the cell membrane itself as they become incorporated into vesicles. Vesicles are essentially liposomes that are carrying cargo. Vesicles bud off of the ER and go to the Golgi. They bud off of the Golgi and go elsewhere. They can go to the cell membrane itself for secretion. And we're going to talk all about vesicle formation, vesicle trafficking, and budding in a much later lecture. But as these vesicles are pinched off of the ER and they fuse with other organelles, well, then the ER membrane becomes part of those other organelle membrane, and these newly made organelles are distributed. So we just saw that scramblase works really hard to keep the ER membrane symmetrical so that we don't get these induced bends, but asymmetry is actually characteristic of most cellular membranes. What does that mean? That means the membrane components on one face or side of the membrane are distinct and different from the other face. If we're talking about the membrane itself, the cellular membrane, what we mean is that the membrane components on the cytosolic face touching the cytoplasm are unique and distinct from the membrane components that are on the extracellular space coming in contact with the outer world. So asymmetrical, we mean not even on both sides, distinctly different. And the inner face of the cell membrane is distinctly different from the outer face, and that's intentional. This is true both because membrane proteins have specialized ends. If we think about a receptor protein, well, the receptor protein should have its ligand binding side on the extracellular end, because that's where these signaling molecules are going to arrive. And it should have its information transmitting end, that's cytosolic, because we want to transmit information into the cell. So membrane proteins need to have an asymmetry, and the lipids have to have an asymmetry as well. The lipid composition is different on the two layers or the two faces of the bilayer, and that is also intentional. We can easily maintain this asymmetry because we don't get a lot of transverse diffusion, and even if we do get transverse diffusion, flipase is going to come and fix it. So the rarity of transverse diffusion keeps this asymmetry in place. If membrane components were allowed to flip-flop from one side of the membrane to the other over an enormous amount of time, but over time we would reach this homogeneity. And we don't, because transverse diffusion is so rare and flipases fix it when it does occur. So the membrane is asymmetrical and it remains asymmetrical. Yes, as new membranes are made by the ER and pinch off as vesicles, those vesicles confuse with the cell membrane, make the overall cell membrane larger. That's how cells grow in size, is by this unidirectional movement of vesicles from the ER and the Golgi to the plasma membrane, making the plasma membrane bigger and bigger, making the cell bigger and bigger. If certain components of the lipid bilayer arrive from the Golgi or the ER to the cell membrane on the wrong side, flipase is going to take care of that right away. Flipase is going to make sure that the correct cellular components are on the correct face of the membrane. So let's talk a little bit about glycolipids. Glycolipids are membrane lipids that have sugars added to them, glycolipids or sugar lipids. These are much more prevalent on the outer face of the cell membrane and virtually non-existent on the inner face. These sugar groups of the glycolipids actually act as a sort of like, I don't want to say cell wall because there are no cell walls in eukaryotic cells, or at least in animal cells. But I'll borrow the language from the textbook. Creates a kind of cell coat that surrounds most of our cells. So our cells don't really have their phospholipids right out there in contact with the outside world. Remember, the phospholipids have the consistency of olive oil. Instead, the sugar groups of these glycolipids are what's projected on the outer surface of the cell, and that creates a bit of a boundary, a protective layer. The protein component of these glycolipids and glycoproteins are made in the rough ER. The sugars themselves are added in the Golgi, and then the entire package gets to the cell membrane by riding as part of a vesicle. So it's a multi-step process. Build the lipid in the ER, put a protein on that lipid in the ER, add sugars to that protein in the Golgi, and then a vesicle brings the entire structure out to the cell membrane. And if it's inserted into the wrong side, flipase will get it to the right side. Other membrane components have other prevalences. For example, large phospholipid head groups, hydrophilic head groups, tend to be on the outer surface of the cell membrane. Small hydrophilic head groups are on the inner surface. That contributes to the spherical nature of the cell because there's more stuff, more volume on the outer leaf than the inner leaf, which causes the cell membrane to bend in the way that we want it to, bend in the way that we didn't want the ER membrane to bend. And there are even other differences between the two faces of the cell membrane, but going into those would be going into much more detail than we need, and so we'll wrap it up there. So to summarize what we talked about today, The plasma membrane is a lipid bilayer. It's got hydrophobic tails in the interior and 2 layers of hydrophilic head groups on either side. And the plasma membrane, the lipid bilayer, separates the inside of the cell from the outside of the cell, the entropy of the world from the order of the cell, and protects the cell, allows the cell to maintain homeostasis. Membranes act as both barriers and portals. The barriers are the lipids themselves. The portals tend to be membrane proteins, and these membrane proteins regulate the passage of molecules in and out of the cell. Phospholipids themselves are amphipathic. They have hydrophobic tails and hydrophilic heads, and because of this amphipathic nature, these membranes will form spontaneously, all on their own, with no energy input needed. The universe striving for entropy is what causes these structures to form. Also, that striving for entropy is what allows the lipid bilayer to be self-sealing and assume a flexible spherical structure, all to keep those exposed hydrophobic tails from being exposed, and to have the hydrophobic tails buried in the interior of the lipid bilayer infinitely, so that water never sees them and can never be ordered by them. Membrane fluidity is influenced by lipid composition, the length of the tail, the degree of saturation, all of that can be regulated by prokaryotes, or in animals by cholesterol content. Cholesterol regulates membrane density and membrane fluidity. It fills in the gaps between phospholipids. It allows van der Waals interactions to form. The more cholesterol you have in a membrane, the more that membrane will be dense. The less cholesterol, the more fluid the membrane. The lipid bilayer is a two-dimensional fluid. Lateral diffusion is fine. Things can largely float forward, backwards, left, or right without any problem at all. But transverse diffusion is restricted. Things don't flip upside down and leave their face. If they do, Flip-ACE is going to come in and fix that right away. New membrane lipids are made in the ER. They're inserted into the outer leaf of the bilayer, and then they're moved by scramble-ACE to evenly distribute them across all layers, keeping the ER membrane from becoming deformed. The cell's membrane, though, is intentionally asymmetrical, where we have distinct lipid and proteins on the outer surface. That's where the sugars are, so we have that nice protective sugar layer, and then the inner surface has its own collection of protein faces and lipids. This allows for the membrane to have the spherical structure and allows for proteins to properly work so that their extracellular domain is facing the extracellular environment and the intracellular domain is pointed inward. Glycolipids are membrane lipids that have sugars added to them. Glycolipids are concentrated on the outer membrane to form that protective sugar coat, and Flip-ACEs will always be used to get membrane components to their right face, to their right leaf of the bilayer, if and when they should happen to transversely diffuse. Overall, membrane composition and fluidity are essential for life. Cells can't survive without the principles we've discussed here. In detail, cell signaling, transport, cellular function requires membranes to have the correct proteins, the correct lipid composition, and the correct fluidity. And without membranes, or without the correct membranes that are ideally suited for what those cells or organelles need to do, life simply wouldn't be possible. So yeah, one came in a little bit shorter than usual. Even the next video this week will be shorter than this one still. So some nice light introductory information about membranes. Hope you liked it, and let me know what you didn't, and we'll talk about it in class. As always, thanks so much for watching. I appreciate it.